So what we're looking at today is some post-transcriptional modifications of mRNA. In our last section of notes, we learned that transcription produces mRNA, or otherwise known as messenger RNA. And so today we're going to talk briefly about the steps that occur here. We'll go into more detail tomorrow. But then we're going to talk about, well, what happens to that mRNA before it's actually completed. So the first thing that we need to know is to understand some portions of the DNA code. Right here you have a molecule of DNA, and within the molecule of DNA you have a section of DNA known as the gene. Within the gene you have several portions. You have a promoter, which is a non-coding section of DNA or section of the gene. You also have the what's known as the coding region. This section would be my coding region. And then you have the terminator, where it basically tells the transcription factors to stop. So one thing that's important to know is that you have a promoter, that's the non-coding section of the gene, and it's upstream of what's known as, or in front of what's known as the TATA box, which actually tells us where to get started. If there's a mutation, the mutations won't attract what's known as our transcription factors. Transcription factors. These are proteins and enzymes that actually allow this process to occur, are kind of our workhorse of the process. So our first step in transcription is going to be known as, number one is initiation. So initiation is the first step, and this is again going to occur upstream of a gene, and what we do is we attract an RNA polymerase, just like how we had a DNA polymerase. What happens is in front of the gene, or in front of the right where the promoter is, we will transcribe after the TATA box. So the promoter can include our TATA box, and after the promoter, this section of the coding region is what we actually transcribe. What is transcription? Transcription is just rewriting the code of DNA into mRNA. So your mRNA polymerase, or your RNA polymerase, will base pair DNA to RNA to create our mRNA strand. But before all of that can be completed, we have a step called elongation. Elongation. And in elongation, we're going to add the RNA nucleotides. Okay? And when we add this, we're going to build, just like DNA, from the 5' to 3' direction. We're adding base pairs. So initiation is actually attracting what's known as transcription factors. Right here, these molecules are known as our transcription factors. And they're going to attract our RNA polymerase, kind of gather the molecules, the machinery together to get ready. And then elongation occurs is when we actually add the bases to make that mRNA. After elongation, we're going to have the step known as termination. And basically we're at the end of our gene. And then our RNA polymerase is released, and our DNA will rezip, close back up, and our mRNA is released. The end result of this process is that you have complementary sequence of a gene that can travel to the ribosome. One of the big reasons why we need transcription to occur is because DNA can't leave the nucleus of the cell. It's too big, it's too important, it can actually get damaged if it leaves the nucleus and goes into the cytoplasm of the cell. And so here what you're seeing is a couple of things. Our transcription factors will help to open up our original DNA sequence. Once it opens and unwinds the DNA sequence, we actually have RNA polymerase adding in the base pairs, and then when we add up the base pairs, we're actually producing our mRNA strand. We will reach a terminator, and that tells our RNA polymerase to stop. Once it stops, our mRNA can be released, DNA can coil back up with helicase. But after that's done with termination, we're actually not done with the mRNA. We have what's known as some post-transcriptional modifications that have to occur. So, some modifications. These modifications are going to actually help regulate gene expression. We have two sections of the RNA code. Two sections of the RNA code are known as introns and exons. Introns are going to be known as, you can think of them as inferring information, okay? And what happens with the introns is they actually get, or they are actually the non-coding sequence that is removed. On the other hand, you also have exons, and your exons are actually what are expressed, the information that's expressed. And so therefore, your exons remain in the molecule of the mRNA. Okay, so you have your DNA, you have your promoter, here's the RNA coding sequence that gets transcribed, here's your terminator where we stop. Promoter start, terminator stop. We go through transcription, so now this is our pre-mRNA. Our mRNA has exons and introns. We cut out the introns. Once we cut out the introns, all we have left are our exons, and also you can see there's a leader and a trailer here. Next, what we have are some other enzymes or molecules known as spliceosomes and SNRPs. The spliceosomes and SNRPs, specifically the spliceosomes, are going to be a multi-component enzyme that removes the introns and it links the exons. So that's the enzyme that actually gets this job done, that makes this happen. Once we remove our introns, this is actually going to be really important for the antibody formation, to actually make our antibodies. And this also allows for something known as alternative splicing, which means that sometimes we actually remove different introns. And depending on different introns that we remove, alternative splicing will actually allow for a variety of mRNA and eventually proteins to be produced. Okay? But once we get there, we also have something else known as our 5' cap, which you see up here, and our poly A tail. Our 5' cap is going to be a series of nucleotides that's added to the 5' end, which makes sense, its name, of mRNA. This actually helps prevent the degradation of the mRNA, so it helps prevent damage to it. It's also going to help for the signal to exit the nucleus. It also is going to help signal to bind to our ribosome. Okay? So our 5' cap is basically like the leader, the cap versus the tail. It's going to tell the mRNA to leave the nucleus, and once it leaves the nucleus, and I need to get rid of that. Once it leaves the nucleus, it can actually go and bind to the ribosome there. And then the final part of the modification of mRNA is our poly A tail. The poly A tail is going to be actually a group of adenines that is added to the 3' end. And again, it helps prevent that degradation of the cell, or of the RNA molecule. So you want to take some time to look over these pictures and relate them to the post-transcriptional modifications. It is important that you know how this is related to the making of different proteins. Different sequences of DNA, different genes are going to produce different mRNA. In addition to that, the modifications of the mRNA can create even more variety and more differences in the proteins that are being made, and eventually in the traits that are being produced.